All right, folks, welcome to Chapter 7 on the cellular respiration business. Okay, so let's get started right away. Okay, the first thing that we have here is just an overview of what does oxidation and reduction mean, and this is right from your book. And if you notice, okay, what we have, okay, I'm on a blue pen here, is we have a description. So basically, oxidation and reduction is simply the transfer of protons, okay? Protons and electrons specifically. So when we're dealing with the molecules in respiration, we're dealing with NADs and glucose, and specifically the hydrogens, the protons, and their associated electrons. So notice, if you're going in this direction, things are being oxidized, and if you're going in this direction, things are being reduced. Reduce is means to add the protons and electrons. Oxidation means to remove them. So if you start off with what we have here, this energy-rich molecule like glucose, and you have an enzyme, it can take these protons, I'll change the pen color here to red. It takes these protons and their electrons from the glucose, and it's going to transfer one of them to NAD+, okay, through this enzymatic reaction. So that's all this is talking about. So when we're talking about NAD and FADs as the carrier molecules, they're carrying these protons and the electrons that go with them, meaning that they are getting, the NADs are getting reduced, okay, because you're getting protons and electrons added to them, all right? And this, of course, over here is your product when you start looking at the cellular respiration. That is then used to transfer the protons and the electrons to the next phase, meaning, for example, from the Krebs cycle to the electron transport chain. So this is just a detail of what's going on. Okay, the next one here. How does this whole process work? Well, you get these great electrons, all right, from the glucose molecule, and they're very, very high energy, okay, and by enzymatic reactions in the electron transport chain, and that's represented here by the staircase, as the electrons literally fall down in their energy levels, that energy is released and utilized to pump the proton gradient so that you can run that ATP synthase molecule to make ATP. So that's what's happening. You can envision every one of these steps as like an enzyme that's using a little bit of the energy that is contained within the electrons to do the pumping of the protons, which we'll get to later. Okay, so that's the idea, the graphical representation of the electron transport chain. Okay, so this is where things are going to happen. So outside here, in the outside of the mitochondria, in the cytosol, okay, that's just the cytoplasm of the cell. That's where you're going to get glycolysis happening. And that's where you take glucose and break it down to molecules of pyruvate, and then you feed a couple of carbons at a time into the mitochondria to go to the Krebs cycle. And that's right here in the inner mitochondrial space. And then, oops, I should probably change the ink color here. And then after that, it's going to go to this membrane, the inner mitochondrial membrane, for the electron transport chain, and that's the final process. So this is just telling you where these things happen, all right? The cytosol for glycolysis, the Krebs cycle is going to happen in the inner mitochondrial space, the matrix, it's called the matrix, and then the inner mitochondrial membrane between the outer space and the inner space is where your electron transport chain reaction will occur. So make sure you know where those three things happen. All right, so what we have here is we have a breakdown of the idea of glycolysis. So you start with glucose, and you're going to phosphate it, and basically you're taking a 6-carbon sugar and turning it into these 2 molecules of essentially pyruvate, which are going to feed down the line here. These are the 2 molecules of pyruvate. You do not need to know that all the enzymes involved, but what you should know is that it takes some energy. Okay, so if I go back to my red color, it takes some ATP. Okay, you'll see that happening here. You have to use a couple ATP, and then look, you get a couple ATP out as well. Okay, so if you use 2 and you get out 4, okay, so there's 2 out here and 1 here, you have a net production of like 2 ATP in glycolysis. Not very many, but you do get a couple of ATP produced. all right? And so you have to burn a little energy to make a little more energy. But the basic idea here is you go from 6 carbons down to 2 3-carbon molecules, okay, called pyruvate. And that's the end product, essentially, of this first part of glycolysis. All right, so now, once you've done that, you're going to take 2 of those carbons, okay, 2 of the carbons from each of the pyruvates, and those 2 carbons are going to be fed into the Krebs cycle. How? They're going to be attached, okay, to this molecule here, which is coenzyme A, and you attach the 2 carbons, this is the 2-carbon unit that you attach to coenzyme A, and coenzyme A is a delivery truck and it transfers it to the Krebs cycle, okay? And right now we're in segment A of the Krebs, and that's again in the matrix of the mitochondrion. It drops off the 2 carbons, and then notice coenzyme A goes away to pick up 2 more carbons and bring them back. So it just shuttles these 2 carbons between glycolysis and the Krebs cycle, okay? And then you don't need to know all the intermediaries. That's not important. It goes around the circle, okay, back to its starting point. So you start with this 4-carbon molecule, oxaloacetate. You add 2 carbons to get a 6-carbon molecule, and then through processes around the Krebs cycle, you lose both of the carbons and get back to where you started from. So things to know. Definitely know what I just circled, oxaloacetate in segment A. Know that you have 2 carbons that are added to it. You don't have to know the names of the other carbon molecules, but what you do need to know, and I'll change colors now to purple, is you definitely need to know that you produce 1, 2 NADHs for each of the 2 carbons that come through, and 1 FADH2. All right, so that we call the NADs and the FADs. The NADs and the FADs are produced here in the Krebs cycle, and they carry. Remember from that first slide, when you reduce them, they get reduced. Notice it goes NAD plus to NADH, and FAD to FADH2. So the NADs carry 1 hydrogen and some electrons, and the FADs carry 2 hydrogen and some electrons. And those are the valuable cargo that are going to be transferred from the Krebs cycle to the electron transport chain. So you definitely need to know that you have oxaloacetate, you've got 2 carbons that are dropped off, and you've got the 2 NADs and 1 FAD for every 2 carbons that go around the circle. Now, you might want to ask, you say, oh, what happened to the other carbon from pyruvate? Remember, pyruvate was a 3-carbon molecule, but I noticed that in Krebs cycle there are only 2 carbons that come in. Well, one of those carbons already was eliminated as carbon dioxide before you even get to the Krebs cycle. And notice that here and here you lose the other 2 carbons as carbon dioxide. So by this point here, by the second carbon dioxide, you've lost all of the carbons that glucose ever drops off into the system. So remember, the carbons are not important. It's the electrons and the protons that they carry. Okay, so here is your electron transport chain. Remember the staircase? Well, here we are on the staircase. This is right here on this inner mitochondrial membrane, all right? And what you see here is, look, there's the NAD, and there's the FAD, right? And they are dropping off, they're dropping off their electrons and protons, and those high energy electrons are bouncing from one enzyme to the other. We have the 3 enzymes here, and look, they're pumping these protons that are being dropped off from one side of the membrane to the other. So they build up this big gradient, okay? So if you have lots of hydrogen over here, and you pump them all over to this side, then you get a lot of hydrogens on that side of the membrane, and then if you look, those hydrogens are going to be used over here in order to make ATP. So just to review, these high energy electrons, and that's these guys right here that are being dropped off, they pass through the enzymes. Their energy allows those enzymes, which are the big blue blobs there, to pump those protons from the matrix to that intermembrane space. See, here's the matrix, and there's that intermembrane space. So you're going from like in here to this place right there, okay, in that intermembrane space. By doing that, you create a gradient, okay, so that you can undergo the process of chemiosmosis, which means that it wants to flow back across the membrane, and when it does, we can use that to turn the motor and make ATP for us. So that's the whole benefit. Okay, so what do you need to know about the electron transport chain is that it uses the high energy electrons to power the pumping of hydrogen ions from one side of the membrane to the other, from the matrix to the intermembrane space, so that you can generate a gradient, and it's the gradient that does all the work. All right, and so here we go. Here's another picture of it. All these electrons here, all right, these electrons are powering these blue, the blue blobs in the membrane there, so that they can pump their protons to that intermembrane space, okay, and create the gradient. So I'm going to highlight in yellow. The gradient is being made in this space here. This is where that proton gradient is going to be. It's going to be high concentration of protons in this space here, called the intermembrane space, that they're then going to want to flow back towards the matrix. And the only place that they're going to be allowed to do that, okay, is going to be here. I just circled it in green, which is our ATP synthase, and that's where we're going to get all that great ATP made. So here's ATP synthase, here's the intermembrane space, and this is the matrix. So remember, we had all these protons here in the space. Now they want to say, gosh, we're so crowded on this side, we want to go this direction. And so, oh, fortunately enough, there's a place they can do that. They can go right through, ATP synthase, and when they do that, ATP synthase turns, and when you turn, you get this happening. You get adenosine diphosphate and an inorganic phosphate group attaching together to make ATP, which is our energy. And a lot of this happens. So this is where you get like 32 of these things being produced just from this particular enzyme utilizing this gradient that was made in the electron transport chain a couple of enzymes over. All right, so this is what I call the payoff step, because you do get a couple of ATP from glycolysis and maybe one being produced from the Krebs, but that's just like tiny stuff. The real payoff is when you get to the electron transport chain, you set up your gradient, and then you use the gradient to pump those protons right through ATP synthase, and every time it turns, it generates another ATP, and you get like 32 of them. All right, so that is the math behind how many ATP you get with glucose. And again, just an example here. So this is your math, right? If you want the total yield, and it's usually anywhere between like 32 and 38. You see it a variety of ways, but anyway, down here, notice it's lots. Okay, lots and lots and lots and lots are produced down here, and so that's your big payoff step that's happening with chemiosmosis in the electron transport chain. And Krebs cycle here again produces a couple, all right, and you get a couple here from glycolysis, all right, but the big payoff step again is at the end. All right, great, thank you.